Hi, this is Miss Slitton, and this is my wonderful combo class of period 1 and 3 getting ready for their last finals, or essay finals. Say hi. Hi. Wait, that took a long time to say. I think it would be a good idea, since your final is tomorrow, the essay portion, would you like to know some for sure's? Yeah. Let me start with, get these in the group share notes. For sure, know how to do a Hardy-Weinberg problem. You got two equations there, right? Know how to use them. Okay, for sure, know how to do a Hardy-Weinberg problem. Are you recording? I'm recording. We're all recording. Okay, so review that. I have reviewed that many times, and there are past reviews posted, and I don't want to spend time re-explaining how to do that when some of you have that down cool. Okay, it's not fair to the other boys and girls. Okay, here's another for sure, besides Hardy-Weinberg. For sure, know how to do a chi-square test and how to interpret that data. That is another for sure. Okay, I'm going to give you one more for sure. Okay, for sure, understand how to identify like independent and dependent variables. You know, how to write a hypothesis, a control, how to do a flow chart, how to justify your setup. Okay, in general, any kind of experiment. So there's three for sure's, right? Okay, a fourth for sure is evolution is a key concept, right? I would know it, okay, because it is a key concept that you need to understand. So those are some for sure's. Got some science practices, got some evolution in there. All right. Now, did you see that last night I posted answers to all the practice questions? Okay, so, and again, it doesn't mean I'm going to lift one from there at all. Those are just good ways to see how well you're prepared, not just for this final, but really what are we ultimately preparing for? Yeah, the AP test. So that's going to help you. The preparation you put in there is going to help you for your exam in May, for sure. The better job you do preparing. Okay, now Unit 3, which is what we're set to schedule, what's on the schedule to review today, that's going to be things like the cell cycle, like mitosis and meiosis. There's a really easy way for me to figure out if you understand mitosis and meiosis, and that's for me to give you some number on a chromosome, right, for a diploid number or haploid number, and say, draw that one cell under meiosis, steps of meiosis, or a step of mitosis. Then I know, boom, whether or not you understand them. You know, if I say draw anaphase 2 with a cell that has 6 chromosomes, a diploid number of 6, can you do that? That would be an easy way for me to check to see if you understand the cell cycle. Okay? Now, on your cell cycle, remember, it's broken down into what? Okay. What is that? Interphase and what? And mitosis, right? Those are the two. And then interphase is broken down into what? G1, S, and G2. And then you know the stages of mitosis and meiosis, and we have what? Cytokinesis. So you just want to recognize what happens in each of those key events, and you want to know about things like that can regulate it, like cyclins, right? And remember P53, later when we learned about cancer, we learned how that can stop the cell cycle. That can cause what? Starts with an A? Apoptosis. Angiogenesis is when blood vessels grow towards cancer tissue. Okay. All right. So, I think that mitosis is one chapter, and then meiosis and sexual reproduction is in another chapter. So steps, significance, mistakes. Remember we talked about things like how does meiosis contribute to variety, right? You know, how to variation. So you could talk about things like that. Then once we learned about mitosis and meiosis, and that brings us into inheritance patterns. And you're talking Mendel. And Mendel had 3 principles or laws. What were those? Law of segregation, law of dominance, and law of independent assortment. And then anything that doesn't follow those laws. You know, if you say you don't follow the law of segregation, that's an issue. And what is that called? Starts with an N. Nondisjunction. That's going to be a problem. Not following that law is going to be a problem. Is it going to be a problem not to follow the law of dominance? No, not necessarily, right? Law of dominance just says that one trait tends to be dominant over another trait, and it masks it. We can think of exceptions to that every single time with about half the people in this room. Who can we say that to? the males, right? Because you only have one X chromosome. Does that sound familiar? Okay, and like if you remember, I think one of the practice essays asked you about dosage compensation, right? And how do you establish that? And remember, what can we do to make sure you have the same amount of Xness in your cells, male and female? Bar bodies, right, exactly. Okay, so so that would be, okay, so we did law of dominance, law of segregation, and law of what? Independent assortment. And independent assortment works if the traits are on different chromosomes, right? If they're on different chromosomes. I can get a big T with a big R or a big T with a little r. When does that not happen? When the genes are what? Linked, okay? Other exceptions to, let's go back to law of dominance. Other exceptions to that are what? Incomplete dominance, right? Epistasis, codominance, what? Polygenic traits, exactly. Multiple alleles just means I have more than one option in my phenotype, right? Okay. So we talked about sex-linked, polygenic, mini genes, right? And then I'm going to stop for a minute. We're thinking this out with our brain, okay? Go for sleep over substance tonight. Because if you reflect back on all the information you know, and if you go for substance over sleep, like you're going to study another couple hours on one topic or whatever, a couple of topics, when you do that, sometimes your brain gets foggy the next day. And then you can't use it to its maximum capacity. And I'm going to tell you, sleep over substance. Go to bed at a reasonable time tonight. Because you'll get more horsepower out of your brain if you get good rest, a good dinner tonight, you're not freaking out tomorrow. Right? You got to sleep. Don't pull all-nighters. I'm looking at you, Max. Are you thinking about it?
Oh, it's going to happen. Yeah.
Max, no. Yes.
What would you consider to be a reasonable time to sleep?
Well, teenagers are supposed to get like 10 hours of sleep.
Yeah.
7 hours.
I mean, get at least 6.
Oh, yeah.
I get that on a normal basis.
Okay. Ideally, 7 or 8, right? Don't pull this like you're up till 2 or 3 in the morning business, so you get up at 4. I get up at 4. Not at 4. Yeah, don't 4:15. Okay, but get sleep tonight, because I'm not kidding you. It makes a difference. You have to realize I've watched people take tests like this for 28 years. Okay, 2 semesters. That's a lot of tests I've watched. And I'm going to tell you, kids that are tired and exhausted have a hard time pulling from your brain because you're just foggy. This is the way it works. You're not going to get your best work at that level. And besides, there's so much. At this point, if you haven't studied a whole section, okay, really, you can make a difference right now, because I just gave you some for sure's, right? I would do the for sure's for sure. And make sure you know how to do that, because that's guaranteed winner, chicken dinner. But if you're going to, don't get caught up in a detail of something and then not get sleep. That's not going to be good. All right? Okay, so anyway, do that. Polygenic, incomplete dominance. What is incomplete dominance? Like red flower, white flower makes what? Pink. Okay, good. And what's pleiotropic? Yes?
One trait influences different, or so like one gene influences different types of traits.
Right, exactly. So you could have one combination of genes, but multiple different phenotypic expressions from that, right? A combination, like a syndrome, where it's a whole grouping of traits. Good. Okay, DNA history. DNA history. We know so many experiments there to support DNA history, right? And that DNA is the genetic material. Think about some experiments you've told me about. What? Hershey and Chase, Avery McLeod and McCarty, Watson and Crick, Chargaff. What else? Who?
Oh, Rosalind Franklin.
Rosalind Franklin. Yeah, Maurice Wilkins and Rosalind Franklin. Okay, these are all like experiments you can use. Just make sure you know what they each do. Okay, the structure of DNA, you should be comfortable with that, and replication of DNA, there's transcription and there's translation. Can you tell me the enzymes for the replication versus transcription and translation? Okay, so I will spell those out. I need helicase, I need SSB, I need what? primase, right? RNA primase laying down primase lays down the primer. Remember, if it ends in ASE, it's a what? Enzyme, okay? And then you need to talk about DNA polymerase 3, DNA polymerase 1, and you need to talk about ligase. If you're going to do transcription, you're still going to use what? Helicase, right? But you're going to use, instead of DNA polymerase, you're going to use RNA polymerase. And it has to be capped and tailed and snurped, right? And then, when you leave the nucleus, so when you talk about translation, you've got to get on the small ribosomal subunit, then the
Big one.
the big one, the large ribosomal subunit. Okay, so just make sure you have that all separated out. Okay, so that in fact covers 4 and 5. Okay, and then regulation of genes. We talked about all the different ways to control gene and gene expression. In fact, we talked about 5 different ways to control gene expression, right? We looked at chromatin, is the DNA available? Epigenetic inheritance, is it getting expressed, right? Is it even available for expression? Then we talked about the biggest control is what? Transcriptional control, right? Can RNA polymerase bind to the promoter? And we talked about in prokaryotic cells are things like operons, right? And then we talked about in eukaryotic cells, it's not so much about the operon, it's about whether you have transcription factors to help RNA polymerase bind to the promoter. Okay, so then we talked about post-transcriptional control, and we talked about translational control and post-translational control. So investigate those parts as well. Now, we had several different potential essays over this chapter. Where would you find those discussed? On YouTube, yeah, you can look them all up. We've already gone over those. So if there's a part that you don't get, go and watch the YouTube just on that particular question, the YouTube. Okay. Now, do you want me to talk any more about any one of these things in particular that you're feeling uncomfortable about, that you'd like a little more work on? Yes?
I don't really get the Avery, McLeod, and McCarty.
Okay, so Avery, McLeod, and McCarty, they were supporting who? What experiment were they supporting? Griffith. Remember Griffith? Remember Griffith with, and remember the way I remember is Griffith Park, and I think about what's running around in the park, mice, right? And so he was shooting them up with bacteria. Remember there was smooth bacteria and there was rough. And remember the smooth was the one that was virulent, killing them, and the rough wasn't. Do you remember that part of that experiment? Okay, so do you need me to go over that experiment all, or are you golden? Okay, so then what Avery, McLeod, and McCarty did, they isolated with digestive enzymes that either digested the DNA or it digested the proteins, and when you digested the DNA, then there was no transformation. Yeah, so they verified that it was that the bacteria was getting transformed by DNA, non-RNA. What else? Yes?
Can you go for sex-linked
Traits?
Yes.
Yeah, sure. So sex-linked traits, things like color blindness is a typical one. When you do any Punnett squares with those, or you're doing a pedigree with those, then you just have to keep in mind that the male will express that trait if they only have one copy of that gene. So what you tend to do is when you do a sex-linked trait, let's do one, okay? So a female is a carrier for color blindness. I could say it like this. A woman's father was colorblind, though she wasn't. She married a normal man. What are the odds that her children will be colorblind? Okay, so a woman, so here's this woman, she had parents, her dad was colorblind. Okay, so that means that this woman's dad, if he was colorblind, he had an X and a Y, right? Here's her dad, big ears and a big nose, okay? Her dad must have had a colorblind trait, which they actually use little b for colorblind. It's a recessive trait, even though normal vision is fine. So her dad had to give her that bad X. If I tell you she wasn't colorblind, right, then what is she? She's a girl, so she's this, right? So she got a bad B from her dad, but she has to be this, right? and I'm telling you she marries a normal male, so if she's marrying a normal male, okay, then he has to be what?
X big B Y.
X big B Y. Okay, any questions or concerns with the setup? Okay, so then you just do your standard Punnett squares. Okay, and where was I putting your females on top? I don't want to hear it. No. Okay, so if I put X big B, X little b, and then for him I put X big B Y, so this right here would be what? Yeah, normal. It's really, in a normal what? Female. It's really annoying because they use the letter B, which makes you think in your head that that is colorblind. They just use it to indicate that gene, okay? So, and then on this one, and if you're gonna ask me, do you have to memorize those letters? No, okay? They usually give them to you in the question what you already use. Just use whatever they give you. What would this be right here? Yeah, carrier. Would she be colorblind? No, but she would be a carrier. What would this be? Normal what, boy? And what would this one be? Colorblind boy. Okay? So if I said to you, what percentage of her children would be colorblind? Okay? Percentage of children that are colorblind, what would you say? Not is, right? So 25% of her children would be colorblind. What percentage of her boys would you predict would be colorblind? 50%, right? 50% of her boys. You know, her first boy, what are the odds that her first boy would be colorblind? 50%. By the way, what are the odds of her second boy being colorblind? 50%, okay? So, and what percentage of her girls will be colorblind? 0. Okay? Okay? Yes?
Could you explain epistasis?
Yeah, epistasis means standing upon. So when you have a trait, oh, sorry, maybe not reply. Okay, so let's say we say the trait big B anything, guinea pigs are black, and little b, guinea pigs are brown. Okay, but then there's another gene. Okay, big C anything, they have color put down. Little c is no color. All right, so to express this trait, if you are big C, big B, what are you going to be? You're going to be black. What other ways could you be black? Big B, little b, and big C, little c. You could also be black, right? So any combination of big C anything, big B anything, you're going to be black, right? How are you going to be brown? Little b, and what else? Big C anything, right? That's the only way to be brown. Okay, because black is dominant over brown. You have to have 2 little b's to be brown. To have a color, you have to have a big C. If you're little c, and then I don't care what your Venus status is, you will be an albino. This is showing you, so it doesn't matter if you're big B, little b, you're still an albino because the big C gene is the epistatic gene. It is standing upon the other genes. It has to say yes to color, and then the big B just determines which color that is. Okay? What else? Anything else? Yes?
Can you go more into depth on pleiotropy?
Yeah, pleiotropic, a really good example for that is like sickle cell disease, right? Sickle cell disease, if you have 2, I think it's HBsA, why can't I remember that? I don't remember what they call it. I think it's A and something else. If you're homozygous for the sickle cell gene, then you will have sickle cell disease. But if you're a heterozygote for it, non-sickle and sickle, then you are resistant to malaria. So that's giving you more than one trait, because your red blood cells could be slightly sickled, so that's one phenotype, but another trait is you are resistant to malaria. If you have Down syndrome, what are some aspects of Down syndrome? Even though you have just one extra GART gene, okay, you will be mentally handicapped, your tongue will be thicker, your eyes are different, your digits are flatter, you have a shorter lifespan, your body's a little bit stockier. So that one extra GART gene plays itself out physically in your body, structurally, morphology, is different, and your brain capacity is different. So that would be an example of pleiotropy. One gene, but multiple effects. Okay, what else? Yes?
We kind of had a question from the potential essay.
Okay, do you want to move to the potential assays that I gave you? Okay, so we're gonna talk to that. So I can address what you do. Okay, go for it. Which one is it?
Number 5.
Okay, so in case you don't have it in front of you, was this the quick, the grid in or the essay? Free response.
It was the one with the photosynthesis, cellular respiration.
Okay, wait, let me ask what you want. The grid in or the grid in 5?
With glycolysis and etc.
Oh, yeah, okay. Yeah. So that one, did you get my note on there? This question takes some time and has several areas where it could go wrong. Did you read that?
Yeah.
Okay, did you not follow what I did? The math?
We just didn't understand why.
Why you got like 96 and we got 27? Like, how did we...
Oh, here's why. Because it says each molecule of da-da-da results in approximately 2.5 molecules, where each molecule da-da-da, right, I'm not done. It says, in A, it says, what is the difference of total number of ATP molecules produced between 3 molecules of glucose that undergo? So you have to have 3 molecules. Oh, okay. I don't know.
That's why we're
Okay. So if we do the math on that, so the aerobic or anaerobic?
both.
Okay, so if we do aerobic respiration, okay? So we get ATP directly in aerobic respiration. Where do we get ATP directly? Glycolysis, how many do we get? 2. Where else do we get ATP directly?
No, you don't get it.
Come on now.
Krebs cycle.
Krebs cycle, which we do, we get 1 a turn, but we do it how many times? Twice. Okay? So ATP directly from 1 molecule of glucose, you're going to get 4 ATP from that. Do you agree with that? You understand why I got that? Okay, so then we need to start dealing with the reduced NAD issues, which they're showing you it gets 2.5 ATP, right? So we need to take whatever number we get of total NADs and multiply it by 2.5 to figure out how many ATP that is, yes?
Yeah.
Okay, so how many NADs do you get from that? I'll tell you 10, okay? So you get 2 from glycolysis, you get 2 from transition, you do 2 turns of the Krebs cycle, so that gives you 6, right?
Okay.
Is that where it got you?
Because
so there's
we were just going by what was in the table.
Yeah.
Okay, now let me tell you where they got you on that, okay? And this makes perfect sense. They tell you for each pyruvate and for each acetyl-CoA or whatever in the table, but it does it twice, right? Transition twice. Do you see? So that's why you had to multiply it out. You don't multiply glycolysis twice because glycolysis happens once, right? And you know you get 2 from it, okay? But you also get 1 for each of your pyruvates, and you get 3 a turn of the Krebs cycle, so that's a total of 6. So it comes out to 10, so 10 times 2.5 is 25, right? Am I doing, yeah. Okay, and then you get 2 FADs, right, because you do it twice, and those are at 1.5, 1, 2, 3, right? So that's where, and I'm going to tell you right now, I made the same mistake. That's why I know what you did wrong. I did the same mistake, and I'm like, why am I getting different numbers here? And I was like, getting pissed at myself. But here's where I mean, I caught that one early. The mistake I made is I did them all I did the FADs by 2.52. I caught that mistake on myself as well. That was really fun. So that one, it's 5. Yeah, so let me tell you, and that's why I put that comment on there. That's a long one. So I wouldn't personally spend a whole ton of time on that one unless you have a ton of time left over. Because it's worth just as much as a multiple choice where you have a 1 in 5 chance of getting it right, right? If you have A, B, C, D choices, you have a 1 in 5 chance. You've got to do the math on here. You have to do more than 5 multiples on here that you could make a mistake on each one of them. So do those when you know, like, oh, that's easy, Hardy Weinberg, boom, shakalaka, done, grid. But if it's a big old calculation, go, I think I'll focus on another question. Now, use your time wisely. Use it to your advantage that it's worth. If these were worth 5 times the other ones, that would have been smart on their part. If you really wanted to focus on math, make them 5 times, the regular score. But they're not. They're worth the same. So treat them like that. Okay? Yes?
Just to be clear, there's going to be grid-ins and free response tomorrow.
Well, it won't look like that for you. It is just, paper. Yeah, for you to respond. I don't physically have you grid in. And I gave you extra room. I know that's a big complaint that you have. There's lots of room. I'm going to tell you there are 10 questions. Some are very short. Some are longer. And they're between 8 and 12 points each.
So scary.
I know, right?
It's intimidating.
Yeah, it is. But well, I don't want you to I don't want you to come to class and see 10 questions and pee a little bit in your pants. I want you to be like, he's gonna ask us 10 questions. Okay, let's go. Be ready for it. Check them off. All right. Well, yes.
Do we get the whole class time?
The whole class period.
And second,
That's scary that it's not that we need the whole class period.
Okay, go ahead. I'm listening.
You said the HSRT is only the most recent, like, 90.
I'm going to tell you right now, I didn't do an HSRT.
Thank you.
I did not do a single highly suggested reading in class. I've given you like golden child on half of it, right? Okay, now if you're listening to this, I'm not kidding. Don't stay up late. You need to get some sleep. It's super important. Love you very much. Important to sleep.